Good morning, sir.
Morning.
Am I clearly audible?
Can we wait for 2 minutes? Just trying to get it.
Yes, sir.
Okay.
Yes, sir.
Can you hear us? Hello? Can you hear us?
Yes, sir. Yes, sir, you are audible.
Can you hear us?
Yes, sir. Yes, sir.
Your mic is mute most likely, or I can't hear it here.
No, sir, my mic is not muted, but we can hear you.
Now?
So you are clearly audible. So we can't hear you now.
Hello.
Yeah, now I can.
Can you hear me? Can you hear me? Can you hear me? So how is
Yes,
it?
sir. But there is echo, sir.
We'll try to get out of that. Now?
Yes, sir, it's
Now there
better.
not be a...
Yes, sir. It's perfectly
Ready?
fine now. Yes, sir.
What happened?
You speak. Someone said speak.
Please test once. Say something.
Good afternoon. Good morning, sir.
We can't hear you.
They can't hear us. They can't hear us.
Whatever you've done, you can't hear. Can you hear us now?
Sir, you are audible.
Your phone is not working, or your audio is not working. One of them is not working. So,
I muted.
okay, now. But not the others. I'm not getting. We are unable to hear them. Now we've
Hello.
said somebody. Yeah, now we can hear you.
Okay. Hello. Raina, can you hear me?
Yes, ma'am, you're audible.
Okay.
That's because I'm.
Sir, can you hear us now?
Yeah, we can hear
Okay.
you.
Shall we start, ma'am? So, shall we start?
Sure.
Okay. So, good morning, all. and I welcome you all to the launch of CSIR-Jigyasa One Day as a Scientist Week. I welcome Dr. Vinay Nandikuri, Director, Center for Cellular and Molecular Biology, CSIR CCMP, Hyderabad. The chief guest of today, Dr. Geeta Vani Raisam, Head HRDG, all the scientists and other members of CSIR family, and all the participating students who have joined us from 37 CSIR laboratories across India. Honorable PM of India in Feb 2025 mentioned the idea of students visiting their nearby laboratories, planetariums, or space centers and spend a day as scientists there. Following that direction, CSIR took this first of its kind national initiative of ODS Week, where the students will be offered structured scientific immersion for a week. This celebration of science is focused on providing students with immersive scientific hands on experimentation, interaction with scientists, and experiencing cutting edge facilities across CSIR. The event was originally planned during 13th to 16th May 2025, but considering the geopolitical situation at that time and considering student safety, the program was rescheduled for 21st to 25th of July. Now, without wasting any further minute, I would like to invite Dr. Geeta Vani Raisam, Head HRDG, to deliver the inaugural address. To brief our audience about Dr. Raisam, she has a vast experience in academia, industry, government of India, and abroad. She's a PhD in biochemistry from IISc Bangalore and has postdoctoral experience in IGBMC France and at NIH USA. Since her joining the CSIR in 2011, she has been handling a variety of roles ranging from open source drug discovery program to heading business development industry lines at CSIR IGIP. Currently, she has been serving as the head CSIR HRDG since 2021. Over to you, ma'am.
Yeah, good morning, everyone. Thank you, Raina, for the nice introduction and setting the background for the One Day as a Scientist. Firstly, warm welcome to all the 37 CSIR labs, the students who have joined us, the Jigyasa nodals, the scientists, and the directors. I warmly thank each and every one of you for facilitating this One Day as a Scientist weekly program. As a scientist. Firstly, warm welcome to all the 37 CSIR labs, the students who have joined us. I am able to hear myself. Somebody can unmute here. Thank you. So going forward, as Raina mentioned, this initiative was actually suggested by the Honorable PM in Man Ki Baat. The unique thing for CSIR is, apart from him being the Honorable PM, he is also the president of CSIR Society. We have
I request all the participants to kindly mute themselves, please.
Yeah. So we have an additional responsibility to follow the recommendations of the Honorable PM. And as many of you know, that CSIR has always had open day, which is inviting the students to visit the labs. But after the Honorable PM's directives, we decided to do in a structured manner and focusing an entire week and opening up the labs to the various students to come and know what is it to be a scientist. I think many of us did not have this opportunity when we were students, but we are very glad that with the cooperation of all the labs, we are able to provide this opportunity to the young students of the country. And within this week, we hope to attract about 10,000 students to visit the 37 CSIR labs across 22 states. And as you know, CSIR has a vast diversity from aerospace to, underwater missions, to healthcare, to chemicals, and you name it, there is every area which CSIR has expertise on. So all those students, I'm very glad that they have this opportunity, and I request you to be engaged with CSIR labs even beyond this. So it is not just an opportunity today to visit the lab, but you can stay in touch with the scientists and the students. And in future, if you do wish to continue the path of science or even know more about scientists, you're more than welcome. And as mentioned by Raina, while we planned it in May, this has to be postponed. And I'm very glad that today we are having this program. And I would like to thank DGCSR, Dr. Kalaiselvi, for encouraging this initiative. Unfortunately, she couldn't join us today. She was expected to inaugurate and she was looking forward, but she has an urgent meeting which has come up, and that's the reason she couldn't be with all of you today. But we also have an event on the Thursday planned with the Honorable Minister, and I'm sure that Dr. DG CSIR will join us that day and interact with the students. And I also sincere thanks to Dr. Vinay Nandikuri, who has agreed to inaugurate the event today and also talk about what is the kind of science he's doing. So you can hear from one of the leading scientists, what is it to be a scientist? Because you only read in textbooks, and I think this is a great opportunity to hear from one of the scientists in the country. And Jigyasa program itself started in 2016-17. Again, it was an initiative started by CSIR because of the interest which Honorable PM had suggested that science should not stay in the four walls, but you should reach out to the students. As a result of this, from the last almost 7-8 years, CSIR has managed to reach almost 10 lakh students across the country. And it has really emerged as a flagship program of CSIR. And we have carried out many activities like the hackathons, the Jigyasa Vigyan Mahotsav, and all the 37 CSIR labs are very enthusiastically taking this program forward. And my sincere thanks to each and every one of you. And I wish the students a bright future and hope they imbibe the scientific spirit of CSIR. Thank you.
Thank you, ma'am, for your kind words. Now is the time for the formal inauguration of the ODS week. I would like to request our chief guest, Dr. Vinay Nandikuri, to inaugurate the event. To brief our audience about Dr. Nandikuri, Dr. Nandikuri is a molecular and cellular biologist who has contributed to delineating the signaling networks in Mycobacterium tuberculosis. He did his master's in biotechnology at the Indian Institute of Technology, Bombay, and his PhD in molecular and cellular biology from the IISc Bangalore. He was a postdoc at Texas A&M University for 3 years and University of Virginia for 3.5 years before returning to India in 2004. In 2021, he moved to CCMB Hyderabad, India as a director where he established his lab. His research interest is to delineate the kinase-mediated signaling networks in Mycobacterium tuberculosis. In addition, his lab is also interested in identifying novel drug-resistant mechanisms, deciphering transcription, etc. He has 85 research papers to his name in international journals of high repute. He's a recipient of NASI Scopus Young Scientist Award. He's an elected fellow of all 3 science academies in India. Sir, we request you to do the needful, please.
Thank you. Can you hear me? And can you see the
Yes, sir,
screen?
you're audible. Yes,
Can you
sir.
see the screen?
Yes, sir, yes, sir.
It's my pleasure to inaugurate this event. Here it is. What happened? Wow. No, Karilo.
Okay.
That's what I'm doing.
This is from website, sir.
Yeah. Okay, I can do it from the outside. It goes to the same thing. It's our that only. Sorry. Not something in the video. It's too late. Not our internet is good.
Did it is
Then why?
will connect mobile.
It's too late. Okay. It was working. Should I run it again?
No, I think it's fine. It's a bit slow. That's all.
कौन बोला?
Auditor executive. Yeah, okay.
Thank you. I apologize for the some of these technical issues that we have had. Um, can you hear me, see me?
Yeah, we can hear you. We can see you. Yeah, please go ahead.
Thank you. It's a pleasure to have this opportunity to inaugurate this program on behalf of CSIR. It's the vision of our Honorable Prime Minister of India, President of CSIR, for kids of your age to have the opportunity to go to labs and see how these labs actually function. This provides an idea of how the research is conducted in laboratories and what kind of equipments that are used for doing this kind of research. And this, as I was saying, CSIR is an organization which has our Prime Minister, Honorable Prime Minister, as the president, and our Science and Technology Minister, Dr. Jitendra Singh, as the vice president. And we are head by Director General, Madam Dr. Kalaiselvi ji, and who is not here today to inaugurate this, but it's, I mean, because she was traveling to for a meeting with the Prime Minister of Andhra Pradesh, sorry, Chief Minister of Andhra Pradesh. So, in the absence of that, I'm doing basically doing this on her behalf. So, I would like to thank you all for participating in this program on behalf of CSIR, on behalf of President, Vice President, and Director General CSIR. What is the concept of CSIR is CSIR has around 37 labs. One thing about this organization is that we have labs ranging from road research all the way to cell biology, chemistry, structural biology. We have labs in various aspects. Everything, every other aspect, every aspect that there is to be covered is covered in CSIR labs. So, in the context of people or students getting an exposure, depending on the lab you go to, you would end up getting a very different kind of a flavor. For example, even in Hyderabad, there are 3 labs next to each other. There is one IICT, which is essentially the oldest lab in Hyderabad, and followed by us, CCMB. We kind of originated from IICT. 1977, and then you have NGRI. There are all these 3 labs. One of them is a chemical technology lab. We are a biology lab. The next one is a geophysics lab. And same way, you go to Delhi, you will have physical laboratory, and then you have IGIB, and then road research. So there are labs, multiple labs working on various aspects of biology to, as I said, in every aspect of science. That essentially it's not like all of you are going to get the same experience. Today you visit our lab, tomorrow you visit the next one, you'll get a different flavor and the different kind of equipment that are used by people to do and address the problems. So I believe it is a huge opportunity and it's a fantastic initiative by our honorable prime minister, which is taken up by CSIR, and now Dr. Geeta Vani, who's heading the Jigyasa program. And this has been kind of launched, and I'm really glad that I had the opportunity to do so. I believe that in the next 5 days, many of you people would end up getting an opportunity to see our labs and get a feel for it. And maybe some of you will turn around and become a future scientist of India, and maybe you'll work for us. We look forward to all these things. I mean, some exciting things happening because school kids, that generally are the ones with different kind of thought process compared to people who are already molded and gone into a different zone. So I believe it's something that I look forward to hosting you people at CCMB, you kids at CCMB, and I'm sure that is the case with every CSIR lab across India. They would be very happy to host you people and give you guys a taste of what it is that we do. Thank you very much. And uh, should I go to the lecture, or are you going to say anything else, Geeta?
I think you can go ahead to your lecture. I think, yeah.
Okay.
Speak about your science and what made you come to science. Maybe
That I will talk about, but I wanted to
that will
talk.
be inspiring to the students as well.
Yeah, yeah. So I wanted to talk to you guys about two aspects. One is my journey, which is going to be one or two slides. But at a later point of time, I'm more keen on talking to you about something that is pretty much the person would know about something called antimicrobial resistance. A bit of basic biology for you people to go and we appreciate what kind of work gets done in a biology lab. Me being a biologist, obviously, that would be where I would focus on. But I wanted to keep it as simple as possible. And in case something is a bit above this, you can always ask me at a later point of time. So let me start it. We make it full screen. You full screen or somewhere. Yeah. Okay. Okay. Today, what I'm going to do is give you a bit of flavor of how one actually ends up being a scientist, which is what I talk about my journey. And at a later point of time, I'll talk about something that all of us kind of know about, germs that have gone rogue and give you infections, give you fever, give you problems, and bring something like a pandemic like COVID. We'll talk a bit about that. So that's the current focus on what we're going to do. Um, Personally, I come from this, coastal Andhra Pradesh. That's where I studied most of my preliminary education till about 1986 happened in coastal Andhra Pradesh. In 1999, I was in various parts. I actually studied in the vernacular language, which is Telugu, and I was till 10th. I was completely in Telugu medium, and at a later point of time, I started. Got into the English medium after 10th, but it was not something that was very easy for me at that point in time. Once I finished my bachelor's in chemistry, I've actually gone around all over India writing. 2, 3 I had written entrances to ultimately get into masters in a biotechnology program at IIT Bombay. So that's when I think kind of my horizons opened up and I started. I had to speak in two other languages, which is English and Hindi. So it was kind of a learning experience which started when I was 19 years old. Um, and next 2 years have been, adjusting with all these things, learning biology, which I never did. When I last learned biology, it was in Telugu. So, first of all, that in itself was a big change. Then I was surrounded by people who are from big cities. That is another change. Over and above, what I'm essentially trying to say is that it takes a while of effort to actually get used to something that you don't know. Eventually, when I finished my master's, I realized that I wanted to actually do research. That is because we had a project in masters which allowed me to work on fungus. And I also went for a summer training in BARC. BARC, you guys know, in atomic this thing in Bombay. There we worked on yeast. These two things made me realize, unlike you guys who have seen labs, I've never seen labs before I went to IIT. That is masters, right? I mean, I've never seen a proper biology lab. I've seen labs, but college labs where you do some experiments and where you know the result before you start the experiment. So uh, in once I started seeing the labs and once I started doing the experiments for real, I started realizing how interesting it can be. And so I decided to pursue PhD. And for that, was looking around. In those days, the number of institutes that are available to us are not many, were not many. And I had actually ended up in IISc Bangalore for a PhD program in molecular and cellular biology, essentially. So that is when I started working with my mentor, Dr. Umesh Varshney. And the next 6 years, I was spent my time in Bangalore, Indians of Sciences, Bangalore. I mean, in fact, Dr. Geeta Vani is my batchmate from that time. We were all, we have done PhDs together. So, who the heads Jigyasa program currently. So, point is that it's a journey which I kind of started getting a lot of interest when I was around 2021. And then I pursued what I wanted to pursue. After finishing PhD, again, there are one or two paths that are available to you. Just close it. Basically, when we are pursuing masters and then a PhD, I started getting a feel for what I wanted to do. But one can go to industry or one can go into academics. I kind of chose the academic path, but the paths are available. You actually pursue PhD, which side you want to go, right? So the point was that I actually ended up choosing academics, did my postdoctoral fellowship at two different places in Texas for 3 years. And then I was in University of Virginia, Charlottesville for 3.5 years. After all these things, came back to India in 2004. I was look. Can people please mute their microphones? Can you please mute your microphones? Can you please mute your microphones? Thank you. So when you are actually finished it, I started my own laboratory. Starting my own laboratory essentially means that I want to have students, I'm going to have trainees, I'm going to have people working for me, which is basically where you change your role from a person who's learning to a person who's going to teach, right? It's a big change, and I decided to work on a problem which is very relevant to our country. Mycobacterium tuberculosis rather is a pathogen that causes TB. You know of TB, right? So it causes TB. That's where I kind of started working in 2004. And I've been and even now continue to work on Mycobacterium tuberculosis. Basically, I look at various aspects of it. Bottom line, it's a bacteria that causes a disease, right? So now let's go to a bit basics of how a bacteria can cause disease, what is there in our cells, and how we actually can combat this. We have viral infections, we have bacterial infection. What's the difference? So what I'm going to focus on next 30 minutes or so is on basics of some of these things. Let me start with something that we all know, our cell. right? You have 10 to the power of 13 cells in your body. All of you have 10 to the power of 13 cells in your body. And each cell has your genetic material or the DNA in nucleus, and you have many other organelles in the cell that do various functions. There are some fantastic videos that are available in YouTube, which you can see to see how cell functions, what is the role of each organelle, and how they actually contribute to making what they make. And this 10 to the power of 13 cells that are there in your body give you top to bottom various things, brain cells to liver to various other organs. So these are the cells that are there in the body, and they are basically have genetic material inside them. Genetic material is DNA, which all of you know. By now, it's pretty much well known that the DNA. How much DNA do you guys have? You know how much DNA we have? We have 3 into 10 to the power of 9 bases of DNA in each cell. And if you start putting it as a string or a line, it will work out to be 6 feet long. So the DNA that is there in one of your cells work out to be 6 feet long. So imagine 6 feet is as a bit taller than what I am, roughly as tall as me. That much DNA you have in each cell as a single line. And all this is packaged into a cell which you can't see without a microscope, right? So how does this happen? This happens by actually through a something called packaging the DNA. You wrap it around your various things called nucleosomes. This is how genetic material gets packaged. So this is how each chromosome looks, and we have 23 such chromosomes in your body, and 23 pairs actually. Right? These are the various small units in which this is the DNA, and what you see finally is the gene. So there are minimally 30,000 genes and a lot more other things that are present in your body. And can you please turn this off? Thanks. So there are almost 30,000 genes that are present in your body. And what we end up getting is eventually whole machinery. So DNA is your genetic material, but genetic material is simply that it replicates and makes copies of itself. That is not sufficient for you to get an eventual output, which is RNA and protein. Proteins are something that all of us know about. And what happens is DNA gets transcribed to become RNA. And then RNA, can you please mute? DNA gets transcribed to make RNA. RNA then gets translated by a factory called ribosomes to give you protein. So this protein is what is essentially functions for various things. They are enzymes, they are present on the cell surface, they are present everywhere. All these, basically all these things, DNA to RNA to protein, is called central dogma. This was discovered way back in 1950s, and since then it's been considered as the fact that present in among all the organisms and all kind of species. But there are exceptions to it, and so those exceptions are something like you can go DNA becomes replicates it, and you can make DNA from RNA. There are other exceptions to it. So RNA can also be the genetic material for all we know. So here is something that is about from DNA to protein. How does it look?
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Thank you. So that's the bottom line of how our cell is. But now we wonder, our body has all these things, but we all get infections. All of you get cough and cold. All of you fall sick, right? There are times you get pretty high fever, and when you go to the doctor, they tell you that, oh, what you have is a viral fever, or what you have is a bacterial fever. So how many of you know what the bacteria and the virus are, right? So what I'm going to talk about is we get actually not only viral, but we also get fungal infections. You may have seen sometimes, like, you get a patch of some dark thing and it's considered a fungal infection. We get fungal infections, we get parasitic infections. If you know malaria, you would know that it is caused by a parasite, right? And also leishmaniasis is caused by a parasite. And then we also get viral infections and we get bacterial infections. So bacteria, the basic difference between bacteria and virus is the following. Bacteria is an entity which can survive on its own. It has all that is needed to divide and become something bigger, or divide the cell and grow. Whereas virus always requires a host. Sometimes we are the host, sometimes animals are the host, sometimes bacteria themselves are the host for viruses. If a virus infects a bacteria, it's called bacteriophage. The virus infections that we get, they're basically, we are the host for those viruses. There are many examples of that, but basically we are also all prone to viral infections. So what's an infection?
An infection preventionist at Cincinnati Children's. And you're probably wondering, what is an infection? Well, a quick explanation is that an infection occurs when germs find their way into a place they don't belong and grow in numbers beyond the level that the immune system is able to defend against. Infections are caused by bacteria, viruses, yeast, fungi, parasites, and other microorganisms. These causes can occur from direct contact with another person that is infected, indirect spread from contact with the environment, contaminated food or water, and contact with an infected animal or insect. Some symptoms of any infection are fever, pain, fatigue, warmth, redness, and swelling at the specific infection site. Depending on the kind of infection, you may have a sore throat, cough, runny nose, or it could hurt when you pee. There are a variety of tests doctors use to diagnose an infection, and depending on the symptoms, different samples of body fluids are collected, like blood, mucus from a throat or nose swab, urine or stool, and spinal fluid. The majority of infections don't require treatment for most people with a fully functioning immune system. However, when a person's body can't handle an infection, Medication is used to help ease pain and fever, like acetaminophen or ibuprofen, and antibiotics of different types. Sometimes an infected person will need to go to the doctor for other treatment, like draining a wound or so they can be watched closely. The best defense against infection is a good offense. So to help avoid infections, have good hand and body hygiene by washing frequently with soap and water or use hand sanitizer. Avoid sharing personal items like combs, toothbrushes, and other things that can't be or haven't been properly disinfected. Clean and cover scrapes and cuts with clean bandages. Prepare food in a sanitary manner. Avoid unknown pets and wild animals. Use bug spray on yourself and repellent when appropriate for household pets to avoid contact with infected insects. Get vaccinated and be aware of infection risks if traveling and follow instructions to prevent infection. If you are already infected, then stay home to rest and recover. Avoid getting other people sick and cover your mouth and nose when coughing or sneezing. So remember, infection can be caused by a variety of different organisms in different ways. There are many different ways you can reduce the chance of infection. Many infections are taken care of by a body's immune system, and others require medical treatment. Contact your doctor or seek medical care if symptoms are not getting better, getting worse, or keep coming back despite treatment. For more information, go to Cincinnati. Hello.
So there are lots of bacteria which cause disease in our body. There are diseases like you get skin infections, you get lung infections, you can get infections in almost every part of your body, reproductive organs to everything. So all these things, urinary tract infection, you have all kinds of things, including food poisoning. You are all of a sudden get stomach upset and you really have an issue for 2 days, 3 days, you're really, really in a bad shape. All these things happen. So there are lots of infections that we get, and they're not caused by one bacteria. They're caused by more than 200 different pathogens that exist around us. And suppose somebody is infected and we come in contact with them, sometimes they transmit those infections to us, and that makes you sick. So it's basically going from one person to the other person is essentially called transmission, right? The same goes for viral infections. Viral infections can only survive, like viral infections that cause viral fever and other things in our body. They are not living outside. It's not like viruses are present on the surfaces. Bacteria can be, but not virus. Virus is always stays in a host. Like if somebody is infected, they can transmit virus to you, not simply that you'll get virus out of environment. It doesn't happen. It comes from host to host. So if somebody is infected and you come in contact with them, you end up getting viral infection. Please mute the microphone. 200 viruses. There are around 200 viruses that cause infections. Again, viral infections can be for every other thing: cardiovascular, hepatitis, skin infections, sexually transmitted diseases, pancreatitis, pneumonia, eye infections. Name it. You have infections happening because of viruses. There are about 200 viruses that can cause infections. Really think about uh, so many infections. You will think viruses and bacteria and all these funguses are our enemy. Not really. Your gut actually has lots of bacteria, viruses, and fungus. These are useful. These are friendly. And then there are pathogens which are not friendly. So they can cause problems, but then you have many other things that that are present in our body. In fact, our body has 10 to the power of 13 cells, and we have more microbiota or microbiome, which is a combination of all these bacteria, viruses, and fungi that are there in your system. They contribute to 1.3 into 10 to the power of 13. Means we are more bacteria slash viruses and other things compared to humans, number of cells wise. So that much we hold in our body. They have a beneficial impact on us. They break down some of the metabolites which we can't otherwise break it down, so that your gut microbiome is a very important facet. And so these are the things that bacteria also do. But that doesn't mean that every virus that comes your way or every bacteria that comes here is good. You have two ends. You guys may be remembering COVID-19 now, maybe like, started in 2021. Last 2 years, not much has been heard. Every now and then people wake up. But really, COVID-19 is something, is a pandemic. It's a pandemic that came into as a pandemic that came like, 4 years ago to 2020 is when it came. And it has killed quite a lot of people, almost 70 million people, right? And 7 million people, sorry. So when it comes to COVID-19, how we were much, much faster now because of the modern medicine, because of the science, the way the science has progressed, made it very, very, very easy for us to fight this infection compared to what otherwise it would have been. We could come up with a diagnosis. I mean, scientists around across the world and even in India, we could come up with better diagnostics and faster diagnostics within no time, and actually cheaper diagnostics also. Vaccines were made within a year, and there were many treatment options that were tested, and some are available for people who actually were infected. And we kept on following this virus to see how the virus is becoming evolving, means virus is changing its shape. It's almost like first time they came without a beard. Next time after a while they come back, with a beard. So they look a bit different. That's the viral, where you were talking about evolution of a virus. There are basically various forms that have come. First one was Wuhan virus, Wuhan variant. As the time progressed, we ended up getting Delta variant. A little bit later, you may have heard of Omicron variant. All these things were variants of the original coronavirus that caused the infections. So during that time, CSIR was heavily involved in every aspect of CSIR. I mean, not only biology institutes, but every institute in CSIR was involved. Every lab in CSIR was involved in some aspect or other related to COVID. We were doing serosurveillance, genome surveillance, sewage surveillance, air surveillance, and we were giving talks to various people talking about what this thing is. So how do we know a bacteria or a virus, specifically bacteria, is infectious? So this was way back in around 200 years ago, that not 200 years, around 150 years ago, Robert Koch has come up with a hypothesis. He basically is a German bacteriologist who discovered that bacteria causes anthrax, septicemia, tuberculosis, and cholera. How did he do it? His concept was simple. An organism, it should be present in every person who's sick, right? And if you can take that organism out, you should be able to culture it outside. It's true for viruses also. You can culture them outside. And inoculation of that culture back into some animal or other actually causes a disease. And the organism can again be found in the diseased individual slash animal. So which essentially means here is the organism which you can take it out, put it out, culture it outside, put it back in, and you again get the disease. So this is what actually tells you that this particular organism, a bacteria or a virus, actually causes the disease. So if you think of it, when you get all these infections, you go to the doctor, right? You are given something or other, or medicines. What kind of medicines do you get? Antibiotics. So antibiotics are basically given for treating bacterial infections. You can see that part very clearly. You have antibiotics. If it is a virus, you get antivirals. And if it is a fungal infection, you get antifungals. And if it is a parasitic infections, you get antiparasites. These are all essentially small chemical molecules which are called drugs. And these are given to us in a particular combination so that you actually get better. You do get better, but what happens we do whenever we go to the doctors is we are given a 10-day course, or let's say 5-day course. Third day, you generally are feeling fantastic and you stop taking it, right? When you stop taking it, your bacteria that is present in your body is not completely gone. It is very much there. And what it does is that the concentration of the drug in your body starts going down, in your serum starts going down. which essentially means the surviving bacteria can become resistant to that particular drug. How do they become resistant? There are multiple mechanisms, but I'll talk about it. See, once it becomes resistant, next time you give the same antibiotic, they are not going to care. They will grow despite the antibiotic. So here is the scenario. So the person which is resistant will be like, I'm immune to drugs, I'm immortal, I'm not going to die, right? The one which is originally was there, save me from the drugs and I'll die. So the whole concept is originally it was dying, and by not completing your course, you made it resistant. How does it become resistant? There are many ways, but one of the problems that world is facing as of now is most of the antibiotics that we already have in the market, there is resistance available. There is resistance bacteria that is already there. That is called antimicrobial resistance. This is a huge problem. You can't keep on developing newer and newer antibiotics. When we don't take it properly, we end up getting resistance. So right now, there are AMR will be a huge problem in about 20 years, 25 years. We can see it will become one of the biggest thing that will kill. The number of people that are killing by AMR will be 10 million. 10 million is 1 crore. That will be per year kind of scenario. So that's a big deal. When you already have something that can cure and we are still dying because of them, is not so great. So nowadays, quite a lot of times people go to the thing, then they'll say, oh, we have a drug resistant infection. We can all make sure that this drug resistant evolution, at least it is a natural phenomenon, but it is slows down by actually taking our course completely, and by not misusing the antibiotics, walking into pharmacies and simply saying, okay, मुझे थोड़ा सा काशी हो रहा है, दवाई दे दो. So it's almost like that, right? How can bacteria or any of these things become resistant to antibiotics? Multiple ways. What are drugs? Drugs are nothing more than chemical entities. These chemical entities, once they get into your body, are processed. If at any step of the processing, there are enzymes involved, if we can make mutations, if the bacteria can make mutations in those enzymes that would not allow the processing, then they'll be resistant. So that's one way. Inhibition of the cell wall synthesis. Here is the resistance here. They actually make mutations in the target enzymes. Enzymes, if these are antibiotics were affecting the target, a particular enzyme, some mutations here and there, now it's not anymore recognized by the drug, right? You can also modify the drug in itself. So the drug drug is a chemical entity, as I said. If you add a methyl group somewhere and that drug is not anymore effective in binding to the target, that is because the pocket has a certain shape and you're adding a methyl group, you're changing the shape of the drug, and drug becomes ineffective. So the other one is you can also degrade the drug. You can come up with ways to degrade the drug. I'm not asked. It's a bacteria which is coming up with ways to do all these things. It can degrade the drug and in somehow just make it into pieces so that there is no drug available. And the final one, which is also a big issue, is like you're pumping in the drug, it'll pump it out, or it won't let it come in. In both cases, you will end up developing a resistance. And a bacteria uses one or all these combinations to develop resistance to the drugs that we are giving. Same thing goes for virals. They also change their targets. They also do various things. So these things are always an evolution. It's almost like we are trying to do something. They have to find a way to defend themselves from that, right? So, there is always a selection pressure, and there are other ways you can do it. And over and above, the genetic material moves from one place to the other, and these are all actually result in something called evolution of the AMR. There are many genes that are transferred from one bacteria to other. So, like we have our own way in our own community to take care of ourselves, they have a community to take care of themselves, and they do it. They do it pretty effectively. So it's an ongoing battle and it's a process. So as we are talking about it over a period of time, scientists have realized is that we are not alone, obviously. What we only talk about human health. Human health is not alone. Where did you get COVID from? They say it's from bats, right? So from bats, it jumped the species and then became, for us, it became a bigger problem. So you have to look into the whole world as a holistic thing. And the concept is called One Health. And basically, what we are talking about is animals also have viruses and bacteria. Infectious diseases come from any corner, and you have environment. If things get into the water, like such as antibiotics and other things, you have bacteria that is present in the environment. It can become resistant, and then they start moving things around. Humans are ultimately the ones we think about and care about, but the reality is everything is interconnected in our world. And the whole concept is we have to start looking at the whole world is interconnected one way or the other. Plant health is important, animal health is important, water bodies, the way they're kept is important. If you have dirty water bodies, that can cause infections. Clean water in itself solves quite a lot of problems. So there are, again, this kind of a ecosystem is going to be different for a village, and that's going to be different for a town, because our environment is not the same. You don't have farm animals, you have pets, more pets in urban context, less of animals that are present, like cows and poultry, right? So the last thing that I was talking about is basically something called human microbiome. I told you guys that we are more bacteria than human. We have 1.3 times bacterial cells in our body. Where are they? They are there in every part of the body. You can see the things, the number of cells that are there in every part. So you have skin, your mouth, your gut, your reproductive organs. You find microbiome everywhere, and they are beneficial to us. And in fact, certain times when you have dysbiosis, there is some therapy called fecal transplant therapy, which is what feces means, and that is kind of given from a healthy individual to a individual who's not so well, and which essentially leads to them getting better. So now we are in a position as scientists. We can take your human genome can be sequenced in about 24 hours for a cost of approximately 70,000 rupees. We can sequence bacterial genomes for around 5 to 7,000 rupees in just about hours time. I mean, in about 1 or 2 days, we can sequence 700, 800 of these bacteria, which essentially means that we are generating more data than we ever generated in our 10 years ago, for example. So where do we keep all the data? There is so much data that's coming out. Biology is not anymore biology. Biology requires every aspect. Our instruments are made by engineers and physicists. They are involved in various aspects. Mathematicians are involved in churning this big data. Chemists are involved in generating all these new chemical entities for as drugs. So there is so much. The whole research world is interconnected. Ultimately, we are all working for human health. Everything is interconnected, and nothing is works in isolation anymore. To say I am a biologist is one say that I know only this much. And but in reality, to solve a problem, you need everybody. You need people from end to end. So you think if computational biologist is not involved, without computational biologist, we don't even know where to store the data that we generate and how to analyze this data. All these things come from computational biologists, computational scientists, computer science people, and various aspects. And this is how our whole world is going to be. Where in about 5 years, maybe upper limit of 10 years, everything is going to be personal, right? Your genome, you will know what you're prone to infections, you would most probably will know. Things are going to change drastically as the time progresses. We also have to make sure that we are not using up all the resources, like such as antibiotic, and misusing them and making our lives difficult or our next generation's lives difficult. So I thought this is a good platform for me to talk about it, is because you people are the next generation. You guys are the ones who need to understand this aspect. What can we do? One, we need to pass on this information to people widely so that the drug misuse goes down. We have to constantly discover newer drugs, which of course people are involved in. There are many institutes across CSIR, India, and world which are involved in. And of course, we have to use the drugs in a proper way when it is given to you. With this, I would say thank you very much.
Thank you, sir. Thank you so much for your insightful lecture. I would like to call upon Dr. Sridhar, scientist in charge, Jigyasa, to deliver the vote of thanks. Sir.
Thank you, Raina. Uh, Thank you very much, sir, for your insightful talk, and it's really inspiring. In fact, that is the main motto of Jigyasa, especially your uh, travel from a coastal Andhra Pradesh, from a Telugu medium school to become a director. That itself is a big inspiration for all the students, and that is the USP of Jigyasa program. Why we conduct the student scientist interaction program. Thank you very much for your insightful talk, sir. So, uh, to start with, on behalf of the entire CSIR family, I take this opportunity to thank DG CSIR Dr. N. Kalaiselvi for her unwavering support for conducting this one day as a scientist program. Uh, then I would like to thank Dr. Geetavani Rayasam, Head HRDG and also mentor for this Jigyasa program, for her leadership, guidance, and motivation to implement this one day as a scientist program. I once again thank Director CCMD Dr. Vinay K. Nandikuri for the inaugurating the One Day Scientist program and also delivering this interesting talk on the infections and giving a very simple, in a nutshell way, how students can understand how viruses play a different, how antimicrobial uh, infections do happen. I thank all the CSI laboratory directors, Jigyasa coordinators across India for allowing students to participate in this one day as a scientist program and taking this entire program ahead. I thank IIT Bombay Professor Norona Pushpadeep and his team for developing the backend for the system and also registration, helping us registration. Getting now we have around 14,000 registrations for the next 5 days. So it's a huge kind of activity for us. It's a one day as a scientist week that we are celebrating across CSIR. So thanks to IIT Bombay team. Last but not the least, I would like to express my heartfelt thanks to all the participating students across India who are watching us live and for your belief on CSIR, for your zeal, for your scientific acumen, for your curiosity to visit CSIR lab and try to understand how scientists work and try to understand how things are happening. Uh, as one day scientist week unfolds, many of the child who walk into the CSIR lab return home with a bigger. We believe that these students return home with a bigger dream and a stronger belief. And the talk of Dr. Vinay Nandakuri is a testimonial for that. And I believe all the CSIR scientists will motivate all the students. Thank you all. Thank you again for your support and wish you all a great experience. And thank you once again. Thank you, sir.
Thank you.
Thank you.
Thank you, man.
Bye. Thank you.
Bye.